Hello and welcome to the Essential Precursors to EPA and DHA group and video interview series. I'm Dr. Wendy Bazilian, Doctor of Public Health, registered dietitian, and host of these conversations where we're exploring the evolving science of omega-3 metabolism and what it means for nutrition, innovation, and public health and planetary health as well. These interviews are part of our LinkedIn community, New Science on Essential Precursors to EPA and DHA. This is where researchers, clinicians, industry colleagues all come together to share and discuss the evolving research in the field. If you're not yet a part of the group, I warmly invite you to join us. It's a terrific forum for researchers, clinicians, health professionals, and industry colleagues who are interested in where this science is headed. So I am delighted today to be joined by Dr. Adam Metherel. Dr. Metherel is the assistant professor in nutritional sciences at the University of Toronto and principal investigator of a research program focused on omega-3 metabolism, DHA synthesis and turnover, and isotope-based methods for studying tissue-specific lipid kinetics. A mouthful, but he owns it well, as you'll hear. He heads up the lab at the University of Toronto, teaching and mentoring other emerging scientists in the field. Adam's work has contributed substantially to our understanding of precursor metabolism, to tissue conservation of DHA, and the limitations of plasma-based measures of omega-3 status. He collaborates closely with Dr. Wendy Bazilian and is part of an international network of lipid researchers. He's authored numerous peer-reviewed papers that are shaping the current thinking in the field. So Adam, thank you so much for joining us today on this conversation.
My pleasure. Thank you.
Well, to kick off and sort of frame today's conversation, because I want to hear about your research, but also your thoughts on where the research is going on omega-3 metabolism, and in particular, the plant-based precursors, ALA, SDA. Most of us clinicians like myself working in public health and with other individuals, including scientists, industry, we tend to think of omega-3s primarily as fish-based when we're thinking of EPA and DHA. Occasionally we'll hear algal DHA that way, but usually it's the marine sources that we think of when we're thinking of EPA and DHA. In your work, however, you emphasize turnover rather than conversion. And I know that's a prickly topic among the scientists who are right in it right now. But can you sort of set the stage for what turnover is, or instead of conversion, or how they go hand in hand maybe? Can you just tell me a little bit about that?
Yeah, for sure. So conversion itself is really the ability to make DHA from those precursors like ALA and that's high in flaxseed or chia, or from SDA, which from some of my collaborations, we see in very high amounts in ahi flour. So really synthesis, or ability to make DHA from those precursors, oftentimes we're talking about the plant-based precursors. Turnover, which in our modeling in our lab includes the synthesis side of things, is really how quickly, for example, DHA in a tissue is being replaced. So it's not necessarily that levels of DHA are changing. We're actually measuring how quickly DHA is moving through those potentially stable levels of DHA. And so one of the ways you can think about this is if there's a set number of people in a room, say 100 people, but the same rate of people are entering as they are leaving, the levels or the number of people in that room are staying the same, but there's a constant influx and exit of those individuals. So that's really what turnover is how fast that pool DHA is moving through that pool.
A cycling of sorts.
Turnover, yeah.
Yeah. I've been talking to you and to Dr. Bazilian and so forth for a little bit of time, but is that concept conceptually new thinking in the last decade that turnover even happens? Or is that something that's sort of been a thought process, but sort of not risen to the core of the conversation?
It's, I would say, new-ish in the last 20 years. I don't think it's not a brand new idea, that's for sure, but it is something that has been more difficult to measure until more recently in some of the equipment and new methodologies we've been able to develop. So certainly tissue-specific turnover has certainly been measured in different models over the past few decades.
Okay. Yeah, that's so interesting because I'm asking a scientist who's doing this. So of course, to you, it's like, no, this isn't new. I've been doing this. But, to us, sort of like it's gotten to a point where we're hearing about it, which does take time. Certainly, there's a lot of things in science in the field of sleep, in the fields. Yes, they've been working on it for a really long time, but now we have things to say, or like you said, the tools or different ways to measure different parts.
I think the work that was being done on tissue turnover, for example, brain turnover 20, 30 years ago was a little more abstract compared to what we're able to do now, which is we're linking it more to dietary intake and understanding how different levels or different sources of omega-3s from the diet are then impacting tissue metabolism. And I think that's a little more accessible to a lot of people. So
yeah, more real world, we can kind of understand that part. I love the 100 people in a room sort of analogy, because that sort of makes something that looks static on the surface become a continuous replacement or dynamic process, that idea of thinking of turnover that way. And I'm hearing that also the conversion isn't a non-issue anymore. It's just one snapshot of a much larger process that's happening. And it's just one measure altogether.
It's a little tricky because some of the modeling we do is over time, so we can kind of measure it over time, but we're still only generating a single value. So there could be a lot of variation during that period, but we are getting much more information by having a rate or a speed of movement of DHA as opposed to just having a level and that's it. If we only have levels, we don't really understand what's going in, what's going out, or at what rate they're going out. But as long as those levels are steady and consistent, what is going into the pool should be the same as what's going out because the levels aren't changing.
Yeah.
Or what's going into the room is staying the same as what's going out of the room, that kind of thing.
And in tissue specific, to press this point and maybe to go to another question, but to stick on conversion because everyone seems to like to talk about it. But for decades also, while that turnover has been coming down the path in your space, lipid metabolism overall, the dominant narrative, still taught to this day, is that humans are poor converters of plant omega-3s. Why did the field focus so heavily on conversion efficiency? What have the newer models shown? I think you sort of mentioned like different tissues or tools, but what have they shown that changes that view that we're now sort of waking up to or not able to ignore anymore?
Yeah, a lot of it really started 20, 30 years ago. There was a series of different human studies from, maybe a half a dozen different groups where they were providing an oral dose of a stable isotope. So these stable isotopes are essentially a label that's placed on a fatty acid, most often ALA, so that primarily plant-based precursor, adding this label that when a human consumes it, you can kind of track its appearance in the blood over time. One of the limitations, of course, with doing human work is we're often and always limited to blood collection. Certainly there's, you can take biopsies, adipose, muscle biopsies, but most of these studies were done just looking at blood levels and the appearance of the downstream, the products of ALA, the plant-based omega-3. The problem with that is you're only measuring plasma. So those models weren't able to take into account what's going on in other tissues. So even though you're measuring plasma, you're not measuring what was actually produced in the plasma and then taken up by the tissues. So those numbers generated just from plasma would fall in this less than 1% of ALA or even dietary ALA being converted to DHA in the body, but they were unable to consider really the rest of the body. So certainly with the information they had, it was a reasonable conclusion at the time, but the model, some of the modeling that we've generated recently, at least in animals so far, we've been able to identify that same really low conversion level in plasma. But when we've considered the rest of the body, that less than 1% conversion actually turns out to be 9 or 10% of the dietary ALA being converted to DHA. So we can't say whether this is still high or low conversion, but it's certainly much, much higher than less than 1%, at least in these animal models, compared to the whole body modeling.
I was going to ask you about the plasma. That was where I sort of jumped in for a second. I'm glad that you continued to say that. It seemed like that was one reasonable place that you could access. One could access, science could access at the time. And now being able to look at more whole body, is that what you're saying? Whole body synthesis or different tissues. So I wonder if maybe we'll get to this a little bit more later, but maybe that explains a little bit vegan and vegetarian diets and how they could be not consuming any preformed DHA and still maintain that as their dominant omega-3 in the brain. Just sort of hypothesizing there.
So I really, there's a number of other pieces of evidence, and I think one of really the strongest pieces of evidence that we have, and it has really become more prevalent in the literature lately because of the increase in vegan and vegetarian diets, is, and I'll focus on vegan specifically because in their natural diet, they're essentially consuming no DHA at all. They're getting all their omega-3s from plant-based sources. Primarily, it's going to be ALA. But even though vegans are not consuming any DHA, the most abundant omega-3 in their circulation, or at least in their blood, is still DHA. So that DHA, there's only one place that it could possibly be coming from, and that's from synthesis from precursors. So this idea that, we're poor converters to DHA, well, all the vegan, even though we're still just talking about levels of DHA, certainly suggests that we're most likely much better at making DHA than we've been led to believe.
And even so, even if it's low, maybe it's enough.
Maybe.
Simply.
Yeah, that's one of the questions that we really don't know. Maybe it is low, maybe it's not enough, or maybe it is enough because generally speaking, vegans are no less healthy than omnivores or fish eaters. So the vegan and vegetarian population generally are considered quite healthy. So the requirements based on health outcomes are much murkier than I think we know.
Makes you think. Well, you've studied the plant precursors like ALA and SDA, which is a little lesser known, I have to say, in the sort of, at least in the dietitian community. It's newer in terms of the biochemistry and metabolism story, but the SDA from sources like pea flour. Can you explain where SDA fits in the omega-3 pathway and why it's interesting metabolically and like sort of how it differs? You've given me an analogy before when we've talked before about how you differentiate them, even though they're along the same pathway.
Yeah, so the pathway itself has 7 distinct reactions. The more linear steps, there's 8 omega-3 fatty acids and 7 reactions. ALA is a nutritionally essential omega-3 fatty acid, so we have to consume that in the diet. SDA is what's generated after that very first reaction. So it's essentially just adding a double bond to ALA. So that step is also generally believed to be the rate limiting step in the pathway. Metabolically, if there's a way to bypass that rate limiting step, consuming SDA would be certainly a way to do that. We know from collaborations that we've had with high flour producer Nature's Crops, feeding SDA can potentially increase the conversion rates to DHA compared to consuming ALA only. And really the way that works is, to try and simplify it a little bit, SDA is essentially closer to the destination. So if you and your friend who live at different places are traveling to the same event or party or whatever, if they're 10% of the way closer and you leave at the same time, they're going to arrive 10% earlier than you. And that's really
as long as you drive the speed limit.
If you're driving the same speed, yeah.
Right. I just teased because I remember a few years back where they were sort of promoting drive the speed and stay safe. I like that analogy. That makes a lot of sense. I mean, that's very real to a lot of us heading to one spot. We're 2 travelers en route to the same destination. It makes sense that if one of us is closer, we'll get there sooner, but we're both heading on the same path, so to speak. I love that. So the distinction then between these is more the product they come from or the food source they come from, not that one plant omega-3 is a single, maybe that plant omega-3s are not a single category, I guess, if that's the right sort of conclusion to come to, and that they come from different sources and maybe we can get them from different sources from diet, but the ALA is essential. SDA is along that path, removing the rate limiting step. So it moves a little bit faster. It starts a little bit further along the path. Just giving some language to that and understanding.
For sure. Yeah, there's lots of different plants that contain various amounts of omega-3s, walnut, I mentioned flax, chia, ahi flower, canola, lots of different options, and they all kind of have varying amounts of omega-3s. So they shouldn't necessarily be all lumped in together because there is certainly some uniqueness to many of them.
Yeah. Well, it's good to know. And the uniqueness to how they fit in a diet. So bringing it broader to a public health or human or dietitian perspective on this, you wouldn't necessarily have all sources or just one source at any one meal. They fit differently. And the dietary matrix is not just the fat itself. So I hear you on that. Thanks for stating some of the sources because it gives us a broad variety to think about in plant-based sources. I wanted to talk a little bit about one of the studies that you led a couple years ago where you compared 3 diets. I think it was the flaxseed oil, ahi flower oil, and DHA, ethyl ester, if I'm saying it right. But could you, a diet switch model, could you describe that a little bit? What was the main question you were trying to answer in that design? And what happened in that? What did you find?
Well, the question really came down to that idea of SDA being closer to the destination than ALA. So flaxseed is very high ALA content. Ahi flower is also very high ALA content and similar, if not a little bit higher, total omega-3, but about a third of the omega-3s in ahi flower is coming from SDA, that omega-3 that's one step closer to DHA. So we wanted to compare how quickly flaxseed oil versus ahi flower oil versus just straight DHA, how they would turn over tissue DHA, how quickly they'd turn that tissue DHA over. So we feed a background diet to enrich the stable isotope that I mentioned earlier, and then switch them to these 3 diets that have low stable isotope levels, and then look at conversion rates. So what we found was that in liver, adipose, and brain, the rate of conversion and turnover in those tissues from ahi flower was not different from either the DHA or the flaxseed oil, kind of the statistically different. I mean, if you look at the paper, the curves that we generate, they look very different, but they're, as far as statistical analysis goes, they weren't different. Whereas flaxseed versus the DHA were statistically different. So we've kind of shown this, or it's a bit of a proof of concept of this idea that if you consume more SDA, you might actually be able to turn over DHA quicker, which may be beneficial for conversion to some downstream anti-inflammatory metabolites that DHA can be further converted into.
Interesting. So it's sort of like it's not a simply plant versus marine here that we're looking at. Yeah.
No, and I don't want to create a fight between the two. It's just, one doesn't necessarily negate the need for the other. There's certainly some potential complementary aspects of each, that's for sure.
Yeah, it sounds like the discovery is that sometimes like sort of isolating or taking out different arms, you make discoveries about what's going on, to the idea of it coming from different sources, fitting our diet differently. But by containing the ahi flower diet, the SDA diet supported the DHA turnover in the liver, you said, the adipose and the brain, from what I read in your study, at the same rate as the DHA, essentially. Was that
statistically equal?
Okay.
They'd still look a little different, but statistically speaking, they weren't different.
Mm-hmm. Would this be a time to ask you, I know that we've conversed about this before, but you told me about like brain turnover versus liver turnover. This is a little bit of a off topic, not off, but it's a one step removed from the study itself. But you gave a great analogy about that to me that made it more understandable about how different tissues may behave differently and what you're seeing there. Could you tell me about that for a second?
Yeah, for sure. I think the best example is comparing the brain to the liver. If somebody is consuming DHA, the levels of tissue DHA between liver and brain actually aren't that different, but the rate of flux or turnover through each tissue is very different. So again, we can use that 100 people in a room analogy. If both the liver and the DHA are representative of 2 separate rooms, if there's 100 people in each room, one room every hour is gaining and losing 20 people, and the other room is gaining and losing 2 people. That room gaining and losing 2 people, that's representative of the brain. The one gaining and losing 20 would be representative of the liver. So the levels actually can be very similar, but the rate through those tissues are actually different.
And that's dramatic. That's a lot.
Yeah, I think it would fall right in around that 10 times faster turnover in liver compared to the brain. A lot of that can be just based on what the tissue needs are. The brain generally places all the DHA into membranes in the cells, whereas the liver is primarily turning over DHA in order to send it out to all the other tissues, whereas many of those tissues want to hold on to it for different purposes. So the liver is like a production tissue, whereas the other ones are a consumption or conservation type of tissues.
Oh, interesting. Yeah. Oh, that's so interesting. I mean, that's interesting to me. So we think of that phospholipid bilayer. That's what I was first thinking of when you're like, certain are trying to hold it into the membrane and others are trying to like be the production center to not fuel in a fuel metabolism way, but fund and fuel other needs and functions in the body. So this might help explain sort of why the brain DHA remains stable across a wide range of intakes, back to that turnover, and why the blood values might change more quickly, perhaps, and why supplementation effects or some of the measurements that are done in blood might be more quickly to change or less constant. Perhaps.
For sure. The brain is really unique in its desire to keep the same levels of DHA no matter what the dietary intake is. And really, based on the literature, the only real way to be able to significantly change brain DHA levels is to be deficient in omega-3s, whereas other tissues you can supplement DHA and increase DHA levels, 3, 4, 5 times. Whereas if you look at those exact same animal models, but look at the brain, the levels aren't changing at all, but the rate through that tissue is increasing.
Okay. Wow. Cool. Well, our body is so efficient and resilient, as I always come back to, like it knows what it needs and wants for the preservation of itself.
Exactly. It definitely knows what it wants.
Yeah. Well, I want to shift into sort of some of the areas that you're looking at, the sex differences. You've started to see sex-specific patterns in DHA metabolism, particularly, I think, in heart tissue. What are you starting to observe there? What might be driving some of those differences? Can you give me a little teaser into that space?
So one of the main focuses of our research program is looking at sex differences in omega-3 metabolism across a number of tissues. One of the most consistent things we found are these differences in how quickly DHA is being turned over in the heart. So it doesn't seem to matter what the animals are consuming, whether it's ALA or EPA, which is that other fish-based omega-3 fatty acid. Females seem to be using the DHA faster. They have both higher levels and they're using it quicker. And there's certainly potential implications for this. We're only looking at this in animal models, but this could potentially explain or provide a mechanism that might explain lower heart disease rates in premenopausal women or females. It's really kind of a neat finding that could lend itself to potential health implications, but again, with the caveat that we are just looking at in animal models at this point.
Yeah, that's really, really intriguing. It's probably a pretty neat discovery. Was that a direct outcome that you're studying for the first time that you observed this, or was that something that came through another study and then you started directly looking at sex difference?
We're really more interested and focused on what was going on in the liver. And then we just kind of continue. We wanted to see what was going on in other tissues. And this was one of the more consistent findings that we found when we started to investigate other tissues.
Yeah. So different tissues, different sexes, again, different turnover rates like we've been talking about. Same nutrients, but different biology.
Exact same diets.
Yeah. And you've also refined some long-held assumptions. And I know you love to challenge the assumptions we've talked about in science, being a scientist, but particularly around the idea of retroconversion and that EPA increases with DHA feeding. And can you clarify what's happening metabolically and maybe correct if I didn't get that right in the questioning?
No, I think that was pretty good. I mean, this kind of goes back even 40 to 50 years, but the idea that when you are supplemented with DHA only, you actually increase EPA, which is a precursor. Classically speaking, it's a precursor to DHA. So increasing DHA, before these supplementation studies, it was discovered that there was this retroconversion pathway where DHA can be sort of converted back to some of those precursors. So that pathway certainly exists. But what we discovered, or what was believed originally, was that there was an increased flux now through this retroconversion pathway, converting DHA back to EPA. With our newer modeling using stable isotope work and this compound-specific isotope analysis stuff, we were really able to show quite clearly that it's not an increased retroconversion. It's actually DHA through a feedback inhibition process preventing EPA conversion to the next step along that pathway. So in a simple way, you can think of it as DHA, or your body saying, that's enough DHA. You can stop. You don't need to make any more of me because I'm getting it from other sources. So it's kind of telling that one step in the pathway, okay, shut down synthesis. We're getting it from somewhere else.
Yeah, we have enough.
We've got enough.
Yeah.
Okay. In theory, yeah.
Yeah, that's an important nuance, I think, in the field that DHA does convert back. So this is an evolving story. One of the things I've tried in conveying to some of my fellow colleagues and what I've been learning from you and some of your colleagues at Dr. Bazinet is that it's not like the science was wrong. It's sort of like the next chapter hadn't been written. We're like in a storyline where the chapters are evolving because the story evolves, the tools evolve, the science, all the pieces. And I don't know if that's right, but that's sort of where my brain sits with. It's how science progresses, frankly. It's sort of exciting.
Absolutely. And I try to clarify whenever I'm talking about this stuff, it doesn't mean what was concluded based on the evidence then was wrong. It's just now we have new models, we have new ways of looking at this, and we've been able to assess this pathway from a different viewpoint or from a different angle. And now with the new methodologies, we're finding new answers that if they had this technique 30 years ago, I think they would have come up with the same answer. But we don't want to just accept the findings from 20, 30 years ago if we have newer models that might be able to better investigate and answer these questions and really kind of continue to evolve our knowledge base.
Yeah. So not a contradiction of earlier work, just like a refinement of sorts.
A refinement, an evolution.
Yeah, and absolutely not just accepting what was pretty conclusive at the time, carte blanche, because life and time and the modern world and scientific minds evolve. So to step into nutrition and public health for a second, I know that some of this is an animal model. Some of this is like this really interesting isotope tracing tools that you're doing in sophisticated laboratory settings. But how might this evolving understanding influence our dietary guidance? And I know that you're not a dietitian and you're not making behavior change and coaching, but from your perspective, is some of the influence, and especially for plant-based or sustainable diets, what can we glean from this? Or where do you think we're moving in that space of nutrition and public health for humans and I guess animals as well?
Yeah, I mean, that can certainly be a loaded question. I think a lot of this research, particularly relating to plant-based, of course, is being driven by the environment, sustainability questions. Certainly the recommendations for fish oil or EPA and DHA intakes, if you kind of apply that to a worldwide population, certainly would not be environmentally sustainable. So as the plant-based dietary guidance is expanding and increasing in the population, I think having this better understanding of how plant-based omega-3 fatty acids are impacting omega-3 levels and omega-3 synthesis and turnover and all this is really going to help support more informed decisions, particularly relating to environmental sustainability and health. Like I said, vegans no less healthy than omnivores, and in many cases might be more healthy. But that's not to say that I think fish oil and omega-3, EPA and DHA still have a very important place in supplementation, but I think we're kind of moving towards not only more sustainable options, but potentially more options that are tailored to an individual's needs based on their health status, based on their sex, their genetics, their whether they're pregnant or breastfeeding or whatever it might be. I think we're kind of headed in that direction, and I think it's an appropriate direction to head into, to have these recommendations that are a little more specific to the individual or subset of populations, for sure.
Yeah, like sort of that precision nutrition idea that people are tackling it from a variety of angles, frankly. I mean, this being one of them. And there is that piece of growing numbers, the population, the sustainability piece, but really who's doing the eating, who's doing the supplementing, what's the life stage. But again, it's more of a unifying message across the board at getting toward that and learning that sort of what you said, that the body, and there's a lot to learn, but the body has its levels that are still being uncovered for different tissues that may imply good health or optimal health, or, the idea that there's different sources and different types that can make for a more precise, preference-based, sex-based recommendation that fits.
Yeah, it's really tricky to do these animal models and then say, oh, this applies to humans. It's so hard to say that, right? But we need to do this work, figure out what's going on in specific tissues. Can we somehow translate that to human intake and human metabolism and then human recommendations? It's a tricky process, but hopefully something that we're on a path towards.
Well, I see that the value of all the types of science and what they do to either bridge or initiate or bring to. Certainly we know the rules of the road, like you can't generalize even across animals. You can't say this would then apply to all animals, but those different clinically relevant studies that are done that can pave the path for what we need next to look at, or what is possible, or how to test those and press. Thank goodness for scientific minds like yourself who can think like that, what is the value of that, and create a defensible stance for why there's relevance to each of them. I mean, epidemiological research points us in a direction too. Like each of them, everything's a randomized controlled trial in humans, a feeding trial, because the reality of that ever happening, we'd be just paralyzed. I don't think we'd ever find anything out. So all of them feed into each other.
For sure. Yeah.
It seems, especially in this field, that this is an important space that you're operating in. One of the things, and you've mentioned it a couple of times, but just to circle back on a little bit, the challenge or the observation, and this is sort of epidemiological of sorts, that really challenges the conventional thinking on what sources feed the diet is the vegetarian and vegan story, no question, especially vegans, like you said, that virtually consume no preformed EPA or DHA, and they still have those DHA dominant omega-3 brains. What do we make of this biologically? If DHA makes up a big part of what's being measured in the indexes for omega-3 these days, or that we know different things about DHA half-life, what can we make of where our needs are based on how our body will take care of us? I mean, is your research sort of showing us that different sources will sort of make up the difference as we're looking at the rate, the inhibiting step, stopping when we have enough, or propelling it further, or starting with one step closer to the destination? How does that all inform us when we look at populations of vegans who have thrived and have been smart intellectual contributors to our world for so long? And we know about that.
Yeah, that's a tough question. It's a really good question. I think when we're looking at levels of EPA and DHA in our blood, they're really good markers of EPA and DHA specific intake. What they might miss is really how the flux through those pathways, the turnover essentially. How quickly are we using the EPA and DHA? Where is it going? All of that. So the EPA and DHA, very good markers of intake, certainly have been associated many times over with reduced disease risk. But again, it's difficult to translate those values to vegans and vegetarians who don't consume any of it, but are also have these lower health risks of certain diseases, cardiovascular disease, depression, whatever it may be. So I think there may be a path forward in the research community to try and tease apart some of that. Off the top of my head, what those studies are, I haven't thought too long about, but there's certainly a large area of research that can be done, I think.
Yeah, maybe it's a step outside of your laboratory, perhaps, but it's certainly one of the biggest trends at the moment is like wearable technology and monitoring devices and monitoring, baseline measures, but recognizing all of them have their limitations. And so are you the right audience or the right patient or person for that tool. Maybe there are other tools or the evolution of things, tools that can help us get a little clearer on that ahead. But that all sort of lends itself to that whole precision nutrition fascination and importance, I think, again.
Yeah, exactly.
Well, we've spent some really great time. I thought I would just sort of ask you about your career a little bit. You've spent a lot of your career sort of questioning the accepted ideas and refining long-held assumptions. You've even told me that that is something that is a drive, something that you enjoy doing. So I wanted just to ask, what drives that curiosity for you? And maybe what excites you most about where the field is heading? I know this is a little just getting to know you better, but sort of as a scientist and what has intrigued you coming in from going into this field, but where you're at now and moving ahead.
It's another good question. It's tricky to answer. For me, I know challenging the synthesis rates, the retroconversion, we're getting into fatty liver and challenging this idea that omega-3s, particularly EPA and DHA, are lower, whether they are or not, in MASLD or fatty liver disease. It's, I think, maybe most predominantly for me, it's just trying to get the answer right. And because, if we're making dietary recommendations on incorrect answers, not that they were incorrect at the time, right? Like based on the evidence provided, but if we have these tools at our disposal where we can refine some of these ideas, I think it's important that we get it right. And that's really what it comes down to for me. And you know what? In 20, 30 years, someone might come along and be like, oh, this work by Dr. Metherel was wrong. That's great. But it's a step in the pathway to getting it right. So I think that's what drives it.
What a humble, but also great answer and contribution. I mean, I think to aspire to get it right is such a noble cause, but also said with such humility that you're one chapter in evolution. I think a really important one. I'm so grateful that the opportunity, sometimes science seems foreign to those who are outside of it. Who is a scientist is a real human being who operates in their day-to-day careers and asks important questions that they're skilled and trained to do. And you've provided such thoughtful and generous conversation around this today. What I take from what you've said today is that the turnover, I mean, my goodness, that seems to matter so much on so many levels, whether it's the rate, the tissue, how we measure, where we're at with that idea of turnover, that not all omega-3s behave the same way, but they're all important. I haven't heard you say otherwise even once, and I haven't seen it otherwise ever. That tissues sort of conserve their DHA differently, maybe, and at different rates when we're, I'm going to keep thinking of the 100 people in a room, and that they're sort of partners along the path, along the multifaceted path. I think that it'll be new news to some that there are 8 steps along the path, because we've so long heard in the mainstream that there's the 3 that we talk about. And
right.
There's more than, but there's the 4 that we've been talking about closely today. And I just wanted to say thank you. So for everyone in our essential precursors to EPA and DHA community, which is a growing community, this kind of work that you're doing, but also not only your ability, but your willingness to talk about it and share what's happening in your laboratory and what you're seeing in the science. I'm sharing some of the nuance and some practical tips, even if that's not your direct day-to-day purview. Yeah. I think it really helps us as a community, and I appreciate it. So I want to say thank you for joining us, Dr. Metherel, today. And I think that that's all for now. I'll look forward to conversing with you again offline, but thank you so much for your time today.
You're welcome. Thank you.